My name's Saif, and I'm a data engineer at Basecamp Research. And it's really great to be here and listening to all these talks is super inspiring. But I wanted to talk to you about what we've been doing at Basecamp Research, which is essentially building what we believe to be the largest biological data set in the world, and maybe talk a little bit about the pipelines that we used to get there. So before I kind of jump into the engineering, which it will be interesting as well, but I thought maybe start with the idea of why. Why would anyone want to do this at all? So it's kind of obvious, actually. Every single thing that biotech has ever produced comes from nature. So across all these verticals, in agriculture, gene editing, industrial processes, medicines, consumer goods, flavors, fragrances, all of these things fundamentally come from nature. There's one example I thought which would just be kind of topical or not so topical anymore, but still makes a point. You might remember during COVID, everyone had to do PCR tests. PCR stands for polymerase chain reaction, and polymerase is an enzyme which came out of a microbe from Yellowstone National Park. And not only did it help contain that particular pandemic, but it's an enzyme which has essentially enabled a lot of the modern biotechnology revolution that we're seeing around us. So now, given all this, there is quite a large challenge that biotechnology has around actually trying to use nature in this context. And that is essentially that we don't actually know anything about nature. If you start looking at public data and think about the kind of hypothetical proteins, kind of think about what actually exists out there, 99% of it is still fundamentally unexplored, in almost every kind of protein class, gene class, organism class that we care about. So that is quite a large challenge for biotechnology. I mean, not that they're doing a bad job. They can do quite a lot with that 1%, but I'm sure they can do a lot more if we expanded that. So the question becomes, how do we actually start to expand this? So at Basecamp Research, our core approach is something called essentially environmental metagenomics. And what this means is we travel around the world, we pick up samples, we extract all the genetic material from the samples, and we sequence what we find in there. We have, as I said, we've done 24 countries and more than 125 expeditions. And we're not just doing this in a scattergun way. We're thinking about biology, we're thinking about habitats, we're thinking about niches, we're thinking about biomes. And in that systematic way, we've actually managed to sample 50% of known biomes on the planet. And we're very much aiming to hit the next 50% as well. So, and the reason why we can do this, and this is super important actually, is the way we operate is every single piece of data, every sample that we pick up is connected to a formal permit. And formal permits are only granted via if the person signing the permit has prior informed consent. So they have to understand absolutely what we're going to do with their data. And then secondly, we have this idea of access and benefit sharing, where if we actually monetize anything that we find from a particular sample connected to a particular permit, there's a kind of flow back of kind of either royalties as monetary benefits or non-monetary benefits if we're operating in a country where potentially capacity building or investment might be a more useful benefit. So it all depends on what we negotiated at the point where we signed the permit. So now we kind of get to the data. So it's we've talked about the why, we've talked about the how, and we've also talked about the how from the perspective of the politics of the situation, because going around the world also has that dimension as well. So now leading into the data that we have kind of three main challenges in terms of doing what we need to do from an engineering perspective. Now, the first thing is what we call about essentially productionizing at scale. And this is a problem probably anyone who tried to work in academia or take academic compute and kind of scale it. A lot of tools in things like bioinformatics and chemoinformatics, they come from research. So they're they're scientifically brilliant. They're designed to solve a very specific problem, to prove a point about data or biology or whatever the application happens to be. But they're not necessarily designed to run at scale. to run for in terms of industrial robustness or whatever. So that's a picture I chose to kind of represent that idea. It's code, it works, it's experimental, it hasn't been tested for edge cases, it hasn't been tested for scale. So challenge one is essentially taking that kind of code and then industrializing it. So you can run the same code, but it's going to run in a repeatable, testable, maintainable, scalable way. All the good things. So then challenge two in this space is more around data formats specifically. Again, staying with the theme of bioinformatics, because that's a large component of what we do. The data formats that exist in bioinformatics are proprietary, and not proprietary in the sense that they're not standard. So that you can't use standard data tools to actually query these things. So across these file formats, you get data silos. It's not very easy to say, can I query data from this sample, this sample, this sample, and ask a question across the whole data set. So the second challenge was essentially moving from these proprietary file formats into a standard database or warehouse type solution. And then the third challenge, now this was also quite interesting, and is essentially because we don't just want to query the data, we want to do machine learning, we want to apply models, we want to train models, we want to fine tune existing models. We needed to build a platform where scientific computing, so bioinformatics, chemoinformatics, physics-based computing, can run, but then create version models and version data sets. So all these processes need to produce data sets, but we need to keep track of the lineage of what they are, where they came from. And then if we're going to train models or fine-tune models, maintain the association between the model and the data set. And also maintain the association of the data set with the scientific workflow that it came from originally. So now I'll just do a little bit of history about how we took this on, kind of almost chronological, or yes, chronological. So I was a very early employee at Basecamp, and when I came in the door, I kind of came out of academia. So where we started, where you'd expect us to start, a combination of Bash and Python scripts. And this worked it worked okay at the kind of scale that we started with. We didn't have a lot of data, and we had a couple of customers, and we could spend a lot of time trying to deal with their problems in a very bespoke way. So we started getting more traction in what we were doing. We started getting more data coming in the door. So our second stab at this was staying within the domain. So we started thinking about specifically DSL's domain-specific languages from bioinformatics. We looked at two things, and we implemented two things. One is SnakeMake, which is like an extension of Python, which is a combination of Python and Make, might be the best way to talk about that one. And then also Nextflow, which is DSL based in Groovy, and Groovy is a Java-compatible language. Okay, so, and then that worked quite well at a reasonable scale. We needed to move on again because more data started arriving. So this led us into the cloud. And essentially, we built something like this, where we were using cloud storage, cloud-based object storage, and we were on AWS, as you can guess from the icons. And we used a combination of AWS batch, lambdas, and step functions to start processing what we were doing. And this actually worked quite well, and it worked quite well for quite a while, actually. But then we got to a point where we were accepted onto the NVIDIA Inception program. We bought a very nice HPC with a lot of GPUs in it. So we wanted to use all that, because otherwise it would sit idle for a long period of time. So we also needed to move on from this to a kind of essentially a deployment where we could still stay in the cloud, also use our on-prem stuff. And we arrived where we are today, which is essentially, it's environment agnostic data aware orchestration, and how we did this is essentially, this is our current stack. So we have a combination of Dagster, which has got like high-level data orchestration, so we can think about the flow of data around the company and what's happening. And then we're using Temporal for more robust, long-running workflows, because we kind of want to make sure that everything that's running will keep running, we'll have retries, we'll have a kind of more kind of stateful execution guarantees. And then what underpins both of those is a Kubernetes cluster. And that Kubernetes cluster is deployed both on our internal HPC, but then also it extends into the cloud. So if we need to burst into the cloud, we can, or we can stay on our HPC. And that gives us quite a lot of flexibility. Okay, and then similarly, from orchestration, the other idea was around data storage. And again, staying with the chronology, we started with those flat files that I talked about earlier. And then we observed that there's the paradigm of graphs seems to come up quite a lot when you talk about biology, because you've got networks of biochemical networks, ecological networks. So it was quite a nice way of thinking about things. So our first implementation was essentially on this stack, which was using Neo4j to represent what we actually had in terms of metadata, organisms, genes, connecting them all in the way that you can represent them in a biologically meaningful way. But then we also had Postgres, which was keeping the raw data, because the raw data coming off these sequencing machines are very, very long strings, and Neo4j is not constructed to handle very long strings. So this combination served us quite well. And now at the same point where we came into the HPC story with NVIDIA Inception, and we started to scale what we were doing considerably, we then moved on to what is our current stack, which is now everything that we had in Neo4j and Postgres is now represented in ClickHouse, with Hex as being a kind of analytic front end on top of that. So that allows us to self-serve, people can self-serve like ML scientists and product scientists. Okay, and then just to kind of lay out now of kind of what we actually have, the data coming in, we have obviously raw genomic data, which is either coming from our internal labs or it's coming from CROs, which are contract research organizations that might do things like this for us. And on the other hand, you also have field metadata, which is, as scientists physically collect these things, obviously you might make measurements around the sample. So you might care about the temperature of the soil, the pH of the soil, whatever else you might be able to measure. So we have these kind of two main bits of data coming into our orchestration and compute framework, which, as I mentioned, is this combination of Dagster, Temporal, running on Kubernetes. And those flow into our unified data storage, which is ClickHouse. Now we have kind of a kind of bifurcation of use cases from our data storage. One is kind of our product workflows, where we're answering a specific customer question. They might care about an enzyme or a particular protein class, and how we would search for that, how we would validate the in silico predictions that we made. That's essentially what we think of as product workflows. On the other hand, we have the kind of ML training, fine-tuning kind of branch of what we might be doing, and producing these version models. And obviously there's an interplay between what happens in product and the inference of the models, because we might use the models to serve our customers. And vice versa, you might use the product workflows to inform the inference workflows. And then finally, what comes out at the end, I went for quite a generic way of describing it, which is a molecular solution, but essentially it's either a protein or a gene or a set of genes with a particular function that solves a particular problem for customers. Now, I'm moving on. So now this is pretty cool. So I'm just going to kind of touch on now now that's the engineering part done. It's like, what have we achieved with this engineering? So in the first instance, we have way more data. So if green is us for a particular protein class, and this is a very commercially important protein class that you might want to be able to produce synthetic versions of this protein class, the orange is what everyone else in biotech has access to. The green is what we have access to. So we can do quite a lot in that space. And this has led us to 3 quite interesting innovations in ML in various parts of protein space. Actually, no, I'm jumping the gun there. First off, this is pretty cool. This is all of public data to date. So this is everything that everyone in the world has collected to date on the orange line. And the green line is us. So the thing is, this just shows when you have a concerted effort to actually start collecting with the vision of actually capturing diversity, you can actually capture a lot of diversity very quickly if you think about what you're doing. Yes. So maybe just ignore the fact that that's floating off the y-axis. That's just because some legacy data didn't make it into the warehouse. Okay. So just three interesting things that we then produced in various parts of protein. First off, GenAI, we released a model called Zyme Control. What Zyme Control does is it allows you to basically prompt with an EC number, an enzyme class, and you can produce quite essentially usable versions of that enzyme. In protein structure, it's very interesting. You might have heard of AlphaFold, which is a model which essentially takes proteins sequence and goes to produces protein structure. So given a string, it produces the corresponding structure. It's a very useful model. It's very kind of, it's almost industry standard now. The application of our data set, so essentially enhancing the data going into the algorithm, not changing the algorithm itself, led to a 6x improvement in lots of protein classes. And this is quite interesting. I was in the talks yesterday where we were talking about is the product that is the model the product, or is the model not the product? In our case, very much the data is the product. And then finally, similar idea. There's been lots of models from Meta and Google and people trying to annotate proteins. Similar idea because we just have more examples in the data. If you look at the two figures here, where we're trying to essentially classify enzymes of various types, our model, which we released called Base Annotate, or also Hyphen, it's been published in, I believe, Cell. I didn't put the citation here. Again, outperformed these various models on these various metrics. And then finally, this is going away from models. This is a really nice case study where we had a partner. They spent almost 2 years trying to solve a particular problem, spent quite a lot of money trying to solve this particular problem. They came to us, essentially a 1 week in silico analysis, and what we found was essentially solved their problem, and also this on the left just is potentially demonstrates that the sequence homology was really low, which is very important for patent space. So you're getting the same function, but you're very, very far away from what has been patented. So that's very useful for partners as well. And yes, and then finally, I'd like to thank the team, the people who do all the work and made all this stuff actually happen. Gus, Keith, Richard, and Alex. Yes, any questions?